Welcome to Labcorp Perspectives Oncology, where we connect on cancer, science, and patient care. I'm your host, Dr. Rebecca Previs, and I'm a Senior Director of Medical Affairs here at Labcorp. Today, I'm joined by my two colleagues, Dr. Kyle Strickland, who's a Director of Medical Affairs and a pathologist at Labcorp, and Dr. Erin Newburn, who's a genomicist by training and a field application scientist who leads our technical portfolio for our biopharma partners. So great to have you both.
Thank you so much, Becca. I'm so excited to be here today.
Thank you for having us.
So let's dive in. Today we're talking about tissue stewardship and precision oncology. And in this era of biomarker testing, I really think this topic is now more critically important than ever. So let's start at the beginning and assume that, not everyone out there thinks about tissue stewardship as much as us three do. But let's walk everyone through various sample types that we use for biomarker testing and oncology. And I'm going to start with you, Kyle. First, let's walk us through why is formalin-fixed paraffin-embedded tissue so widely used right now in clinical and research settings?
So this is for historical and practical reasons. In routine clinical care, anytime anybody has a biopsy, anytime they have a resection, that specimen has to go through a pathology process so that the pathologist can review it, the gross specimen, so that they can identify those areas necessary for a proper evaluation, such as margins, the tumor itself, and any other things that might be necessary for the staging or clinical care of that patient. When that happens, they make formalin-fixed paraffin-embedded blocks. The thing about it is when you treat these tissues with formalin, that fixes the tissue for quite a long time when you combine the paraffin embedding. So these are samples that are archived. We consider them part of the medical record, and they last for years. Now, these aren't the only specimens that we use in medical care. I mean, we have, blood draws. We have different types of tissue that's taken maybe fresh and submitted for things like flow cytometry or cytogenetics, living cell assays. You can even freeze the tissue and look at that under a microscope too if you need a rapid evaluation. But usually those specimens still become formalin-fixed paraffin-embedded blocks later on because they're considered that important. So we have all of these different avenues for looking at the tissue, but what makes FFPE blocks sort of stand out is the fact that they have a very long shelf life and they represent the most important parts of the patient's tumor samples.
No, I really appreciate that. Kyle, for a little bit of disclosure, has taught me everything I know about pathology since we've shared a lot of time at the microscope and research and clinical care. So thanks so much, Kyle. And Erin, I think you have a very similar perspective, but thinking more about this from the biopharma lens. FFPE specimens that Kyle so wonderfully described, are those that are typically available for our biopharma partners. But, their objectives are going to be a little bit different. Tell us a little bit more about that.
So for research, and it's similar to what Kyle just mentioned, it is a very dependable format. It preserves that tissue architecture, the molecular integrity for really long-term analysis. And, if done correctly, this format can, even preserve that tissue sample for decades. And so, the FFPE format that is obtained, it could be a tumor tissue resection that's obtained from a patient, or also a core needle biopsy, fine needle aspirate. And, as you probably would guess, both a core needle biopsy as well as a fine needle aspirate, they're a more limited amount of material. But FFP samples are really quite valuable. They enable really a wide range of research applications in oncology. They can be used for biomarker discovery, biomarker validation, assessing genomic mutations, other molecular signatures from that tumor tissue. And really what happens is often these samples are being used in a retrospective type of studies and research. So perhaps they've been archived for some time, and so they can be stored and then used for later analysis to enable investigators to correlate the molecular data from those samples with the long-term clinical outcomes. And so applications and research, it really varies. Investigators might be wanting to understand tumor heterogeneity in a particular cancer indication, or develop a genomic profile to try to better stratify patients or predict response to therapeutics. So FFP specimens are really a very critical part of translational research in oncology.
Thank you so much for that, Erin. But as a follow up, let me ask you this. We always say there's no perfect test, everything has its limitations. And maybe we can say there's no perfect sample type, if I could offer that. FFP samples, although maybe they're our gold standard, I'd probably throw that out there. They can still be challenging to use for NGS compared to either fresh or frozen tissue. Walk us through some of these limitations and challenges.
Fresh frozen tissue, I would say it's ideal. It could be like a benchmark for molecular analysis. And that's really because if that sample is fresh, it preserves the DNA, the RNA, and really its native state, which means you can get very high quality nucleic acids that are perfect for assays like next generation sequencing or other more advanced assays. But the downside is, a fresh frozen sample, it really requires specialized storage, handling, and unfortunately, it's just very impractical for a routine clinical workflow. So while a fresh specimen, it's ideal technically, it's just not often feasible operationally for testing. And on the other hand, here with FFP samples, it allows that long-term preservation, and there's been very well-established protocols now, and it makes them so invaluable for retrospective studies, biomarker validation. But the catch is the fixation process. It does introduce chemical cross-links. It can degrade the DNA and RNA over time, and that means that the quality of those nucleic acids can be variable, and sequencing and the downstream genomic analysis of that NGS data can be quite tricky. You could, think of it, maybe a good analogy of this, working with FFP samples for NGS testing and the downstream analysis. It's like you're trying to complete a puzzle where some of the puzzle pieces, maybe they're bent, others have faded colors. You can still see the picture, but it takes a little bit of extra effort to try to fit those pieces together.
I think that's a perfect analogy. And you mentioned quality several times. There's so much that goes into the quality of an FFP specimen. So maybe, Kyle, I'll throw it back to you. In the clinical workflow, when we're receiving slides or blocks, what factors are going to influence the quality of FFP samples for molecular testing?
By and large, the most important factor is going to be the tumor content of the specimen. And we have I wouldn't even say that there's strict requirements. We have a limit of detection of around 20% tumor content, 100 to 200 cells per slide. Anything less than that's kind of like the bare bottom, and that'll give us a result. It won't give us the best result, but it will give us a good result for S and V, something that's very reliable. Anything less than that is going to be very difficult for us to examine. And you can imagine that for some tumor types that's easier to achieve than others. For instance, lung cancers are notoriously difficult because you're relying on a transbronchial biopsy or an EBUS procedure, something where they're inserting just a small needle and trying to get a very small lesion in a tumor type that isn't very amenable to getting like very good tissue easily. So, by and large, it's tumor content. Other things that can impact are, whether or not the specimen has been decalcified, because those strong acidic reagents can degrade the DNA. Tumor necrosis is also a factor. If that's kind of long-standing, that can also interfere, just because the DNA is degraded to a point that our NGS methods aren't very good at, and can get clogged. That process isn't very easy when there's a lot of necrosis present. That's it's interesting. So the quality of the DNA, Aaron's absolutely right. In research settings, if you have fresh tissue, if you have whole cells, those can be very easy, and you don't have to have the whole workflow that requires formalin fixation for a set amount of time, and then on top of that, a paraffin embedding process. But even after that, there are additional costs because you have to cut into the block itself, and that histology step can take a lot of time, to be honest. And when that's done, you actually have those sections. They're cut very thinly, and that also means that the cells are cut very thinly too. So you'll see in NGS, some of the best specimens are actually smears, where they immediately take what they've drawn out on the needle, put it right on the glass slides, and then that is the archival tissue that is then used for NGS. Those are whole cells. It's some of the best DNA that you can ever get. So it's not simply just the process of like the formalin is making it degraded. It's like there are so many different factors, and then you're shearing the DNA when you use it on a microtome. I think that what comes back to the pharma side is just the ease of use. You cut it out of a mouse, you can freeze it at -80, and then you can just like scrape off a few cells if you need it, or grow it, however you want to do it. Once it gets to that formalin fixed paraffin embedded step, there's a lot of technical overhead that's necessary to process it further.
And maybe we'll ping pong back to you, Erin, from a research standpoint. What do you feel are the biggest hurdles when working with FFP samples?
I think the hurdles in research, and they're similar to the clinical, testing and some of the topics that Kyle just mentioned. But if I would have to say what is the biggest hurdle, I would say that in research settings, you're tending to work with very limited materials. The remaining materials after all the previous testing, diagnostic testing has been performed. And as we previously mentioned, the fixation causes fragmentation of the nucleic acids, which can significantly reduce the quality and quantity of materials that can be extracted from these materials that are already, very limited to start with. So I think amounts of material available is really I would say, the biggest challenge in research.
But there's certainly strategies or technologies or workflow innovations that have really emerged quite recently to improve FFP samples, that quality, that pre-analytical step, to really try to overcome these sample challenges. Can you share a little more about that with us?
Especially over, I would say, the past decade, there have been many strategies taken. First of all, the amount of tissue we just talked about, that's very limited. One way to maximize the data, the information that you're able to obtain out of those precious samples is to perform a dual extraction of both DNA and RNA. So you can simultaneously extract DNA and RNA from that FFP tumor tissue, which then can allow you for more efficient use of that, again, very limited tumor sample. And you can then utilize both nucleic acid types in your analysis plans. And I guess, another way to kind of mitigate that issue of limited material, limited FFPE tumor materials is really working, to maximize the data you're able to obtain by at the start, from the design of your strategy, design of the assay that you're using in research, utilizing broader genomic testing strategies like comprehensive genomic profiling. It enables you to look at thousands and thousands of potential SNVs or hundreds of genes and biomarkers instead of just, using a single analyte, single gene approach. And so you can utilize these broader approaches like targeted comprehensive genomic panels or whole exome or whole transcriptome solutions. And I think maybe, one last point to this, historically, RNA-based NGS has been thought to be very challenging from FFP materials. There's some more unique complexities for RNA. RNA is definitely a more liable molecule. And so compared again to DNA, but strategies have emerged, and one strategy is the enrichment technology that's used in the sequencing library. They've been optimized. They utilize hybrid capture approaches. So even if that sample has been, very fragmented RNA due to the fixation, utilizing probes that have been very efficiently designed to like tile across that exonic region of the gene, it can enable very, very consistent, high quality sequencing data from RNA. So this enables, very valuable research, ability to look at gene expression metrics from a tumor specimen, or maybe, perform differential expression analysis in your oncology biomarker research efforts. So there have been, I would say, in the past decade, a lot of strategies to try to mitigate some of these challenges.
And I think you've both beautifully described that FFPE samples have been the workhorse for NGS testing for as long as, NGS testing's been around in precision oncology. But now I'm going to ask you maybe an unfair question to look into your crystal ball and try to predict the future. And I'll start with you, Kyle. Do you think that FFP samples will remain that standard for tissue preservation in the era of precision oncology?
So that's a great question. And, I think that when we talk about most of the current tests, the answer is yes. Because, we've developed workflows that can utilize FFPE in these next generation sequencing platforms. We're getting good results, consistent, reliable results. But the other thing is that FFPE is used in so many different settings. It's used for, primary diagnosis via H&E, primary diagnosis immunohistochemistry, immunohistochemistry tests that are coming out that target or determine eligibility for these ADCs and new chemotherapies. I don't think FFPE is really going anywhere, but I don't know if it's going to remain the most important or the only specimen. I can envision a future where we have some live cell assays, and that though and we test each and every tumor against a wide variety of chemotherapies to see what it actually responds to. Right? If something like that were to come around that was cost effective, then that might be just as important. But I think that if we look at the current state of technology now, I think that FFPE is the way to go.
Yeah, certainly here to stay. Erin, maybe from your perspective, do you think researchers or clinicians sometimes underestimate how these pre analytical factors or how fixation actually will impact that data validity?
I would say yes. I think the impact of fixation and some of these other pre-analytical factors is often underestimated or maybe not fully appreciated by both researchers and clinicians at times. And often, as I'm speaking in the field with customers, investigators are, they're focused on how many FFPE slides do I need? How many nanograms of DNA or RNA is needed for this particular genomic assay? And, answering that question is not always so straightforward, as it's not just the quantity of material, but often the quality of the material that needs to be assessed. And so, with the fixation process, if tumor specimen are not prepared properly, we know you can end up with, severely fragmented DNA or RNA, and that means the data you get from the NGS assay might not truly represent the tumor biology. It could be, skewed by artifacts. But, all is not lost. There are so many pre-sequencing optimization factors for testing that you can consider to try to ensure that you can obtain quality data, quality results. And I think we chatted about some of these. Kyle mentioned tumor purity, necrosis, there's specimen storage, fixation times. And so it can be tricky. There's many factors, and they're also kind of invisible at first glance. So histologically, the sample might look great, but molecularly, it's compromised. And so when researchers or clinicians don't account for some of these critical pre-analytical variables, they may risk misinterpreting variants or maybe missing critical biomarkers. And that's why, standardized workflows, rigorous quality control steps are so essential. It's not just a technical detail, but it's really the foundation for generating data that's reliable, data that's clinically meaningful.
And all these technical details matter so much. I know we all interact with people who use our tests on a daily basis. And I've heard you, Kyle, reflect on some of the feedback that you've heard about misconceptions around sample type, FFP in particular. Maybe you could share some of that with our audience.
As far as like the actual specimens are concerned, it's important to realize that those formalin fixed paraffin embedded blocks, like these aren't cells that you can do a lot with. They are fixed kind of in time and space, right, to how the tumor was when it came out of the patient. So we're really limited to what we can evaluate. The DNA and the RNA state at that time, the protein state, the architecture. These are things that the FFPE allows us to do. NGS, the main sort of misconception that I encounter, especially when you compare clinical to biopharma, is the fact that, all of these tumors, they look different under the microscope depending upon the different subtypes. So it's not enough just to look at a primary cancer or to do like pan tumor assays. I think that we need to graduate from that, look at specific tumor types, specific NGS, like, specific gene targets that are determined by NGS, rather than having this whole breadbasket approach, we really need to dive in and sort of pick them apart just a little bit more. They say in pathology there are splitters and then there are lumpers. Well, I'm kind of a splitter, I'll be honest, but we need to split these tumors and kind of look at them individually to figure out what's going on and how to cure them.
So even though you're a splitter, do you think there should be more universal standards, though, for assessing FFP sample quality?
In my point of view, I think that the current setup with laboratory developed tests, I think that that's actually it gives a lot of flexibility to each of the labs. It allows for innovation. So we already have sort of a technical limitation that gives us a standard, that being the 20% tumor content, the 100 to 199 cells. Like that sort of minimum does create a standard in and of itself, but that's based on the limitations of technology. As we get better sequencing methods.
What about you, Erin? Should we implement more universal standards for your customers?
I think there should be standards. There should be recommendations, guidance for assessing FFPE, the quality that's needed for that particular NGS test. I mean, of course, lack of consistency, across platforms can create variability that could impact the reliability of results, and this could ultimately impact, patient results, patient care. So I think in general, quality standards help provide clear, objective metrics for determining, whether that particular tumor sample is suitable to proceed with the downstream analysis. So Kyle mentioned some of the metrics we can use. You can use, minimum DNA yield obtained from the tumor sample, tumor purity thresholds, and other metrics. But, implementing standards, it's not always easy. Different platforms, especially different NGS platforms, have different tolerances for sample quality. And so, I think though the broader objective is just making sure we have accurate, actionable genomic data, and harmonizing some of these quality metrics feels it's essential, because it is what we've seen in other areas, I think, of molecular diagnostics, where there's some level of standardization needed to help drive confidence, but flexibility will always be needed. I think for specific use cases, a baseline of criteria is also needed to really reduce the risk of, false negatives or inconclusive results.
So if we had to change one thing, what would you say, Kyle?
Well, I think that, we're improving day by day. And I think each laboratory has their own sort of improvement processes. And we're way better than we were 10 years ago in terms of like the number of targets, in terms of how we're doing our workflows and our efficiency within the lab. If there's one thing that I would change, I would want to sort of make it more standard that clinicians and oncologists, that they include their clinical notes when they send their samples to us, because there are just so many cases where we have to reach back out. If a patient, I'll give you for instance, if a patient has like a bone marrow transplant, that comes up as a DNA contamination. That's flagged for us, and we have to do sort of a deep dive, and it sometimes creates patient delays when we don't have that clinical information. So I think normalizing, sending clinical notes and keeping the pathologists at these central labs more up to date, I think that could go a long way to improving patient care. And that's not something technical in the lab. That's something just an everyday practice that we could adopt that would help us.
No, I love practical advice, and certainly something that would be easy to implement, honestly, today. What about you, Erin? What would you recommend as a change to current practice?
I think one optimization that would be beneficial in the research setting is just to really try to take a study specific approach in your design, a more precise approach when you're designing the research study. For example, considerations, keep in mind for certain cancer indications. So tissue composition, characteristics of certain tumor types are maybe more challenging to extract high quality nucleic acids from, for example, FFPE. So tissues like breast tissue, they have a high adipose content, or certain tissues that are more metabolically active, like liver or pancreas, they have high levels of nucleases, and these enzymes can degrade the DNA and RNA. So just being aware of some of the specific challenges or considerations for the distinct sample type that you're working with in your your research, I think is important.
And I think that advice could probably be applied to what I think Kyle is going to say to this next question about advice to hospitals, health systems, institutions that are really trying to balance, diagnostic preservation of tumors with research innovation. What advice would you give on that, Kyle?
One of the things that you can do to enable research also sort of enables the NGS analysis in the first place. And that's for pathologists who are reviewing their cases to identify best blocks proactively. And so many institutions that we've encountered, some of the most successful ones, the pathologists sort of have a standard system. Now, whether that be like they have a written report and they stamp something that is then transcribed, later on down the line, versus, you're in the EMR and you actively put in, the best block is D1 representing, the tumor normal interface or something along those lines. Just identifying that block number can go a long way to prevent patient delays. Because otherwise, if you think about it, that specimen gets processed, it gets archived. They have to pull those slides out. Somebody has to actually get the slides. The pathologist has to look at the entire case again to identify that best block. That creates significant times, and we're talking days. And we really we and days really matter sort of in this setting. So I think one of the practical ways that people could enable this is to just be more proactive and have the pathologist document those best blocks ahead of time. And that'll also stimulate research too, because then you don't have to pull all of these slides. You can say, oh, we want to do this experimental IHC on these different tumors. Oh, the best block has already been identified, and it comes out in a nice Excel spreadsheet when we do this query. Like, that's the best case scenario.
I really appreciate that. Well, we've talked a lot today about tissue being the issue, but also, we rely so much on FFPE as our workhorse for NGS testing. How do you both see the integration of FFPE based NGS testing evolving as we move forward in clinical decision making? Maybe I'll start with you, Erin.
What immediately comes to my mind is clinical trial settings and how using FFP tumor tissue may be evolving, or I think is already evolving. And so there are limitations of a tissue only approach in cancer clinical trials. And so relying solely on tissue-based assays can introduce challenges. It can really limit patient eligibility. Many patients may not qualify for a clinical trial using a tissue-only approach, again, which would result in limited enrollment. And I think liquid biopsy testing, so utilizing cell-free DNA testing from a patient's plasma sample, is helping, really driving precision medicine forward. So the liquid biopsy can address many of the limitations of a tissue-only approach in a clinical trial. And I think one of the biggest advantages is expanded patient access. So liquid biopsy enables testing for those patients who lack sufficient tissue for biopsy. So I think pairing both FFPE tumor tissue and liquid biopsy is increasing as a strategy for clinical trial assays, and really stemming maybe from that point, one additional point, as we think more about specific liquid biopsy applications like determining molecular residual disease and instances of earlier stage disease or after definitive treatment of curative intent, that FFP tissue is often used in tumor-informed approaches for molecular residual disease assessment. So that tissue is basically used to help create a somatic signature and then interrogate the patient's plasma to determine, as we monitor these patients following therapy, was circulating tumor DNA detected or not. So basically, was cancer detected or not? And so I think MRD applications, they're quite exciting. There are, many clinical trials underway assessing MRD as an early indicator of patient response. And, we know, unfortunately, cancer is not static. And technologies, assays that allow for these types of temporal insights like MRD testing, I think are going to be in the future really crucial in testing as, we're testing a evolving disease like cancer. So I think we are really seeing now how FFPE tumor tissue and liquid biopsy specimen are really working together and really can complement each other.
I think that is absolutely spot on. And Kyle, maybe I'll turn it to you. How do you see all of this evolving?
When I think of it, I think that we're building for the golden age of genomics. I don't think that we're there yet. We're looking at small panels right now, only 500 genes, and of those, maybe like, a dozen or 2 dozen have really important, meaningful clinical, therapies associated with them. What we're doing is we're kind of setting the stage for whole genome, whole transcriptome, which are just these massive sort of data dumps, right? And it's not until now that we've had sort of that we've developed the tools to assess these. So now you see all of these different studies coming out about machine learning and AI, and the question at first was, how are we going to store all of this data? And then the question became, how are we going to analyze it? And it's going to be through these computational methods that we're actually able to derive meaning out of the genomic, alphabet soup, right? That we're collecting on all of these patients. So we're really building for a better future state. But as this evolves and develops, we're going to see more therapies, we're going to see more targets, and we're going to see an increased ability to detect those.
And what about advice, Kyle, for those audiences out there who may be thinking about bringing this up at their own lab or perhaps in their institution or hospital, just really starting to think about implementation of NGS? What advice would you give?
So my advice would be to don't think that you have to reinvent the wheel. There are many companies, many products out there. Labcorp has their own that will help you set up NGS at your institution so that you can bill for both the professional and the technical component. There's a lot of groundwork that's already been developed. The real value here is that companies like Labcorp have taken the time and effort to sort of get all of this in place, and they're able to get these tests up and running within just a few weeks. So my advice would be, don't reinvent the wheel. At least scope out the ability to use what we've done to help you get to where you want to go.
Thanks, Kyle. And what about you, Erin? What's some advice you would give to our listeners?
Maybe just start with thinking about the basics, focus on, if you are looking to implement NGS, focus on sample quality, focus on pre-analytical factors. We just talked here about all these challenges and complexities of FFPE, but invest the time in understanding the protocols for fixation, storage conditions, because really the poor quality input is what can potentially compromise everything downstream. And validate your workflows, think about these appropriate, post sequencing, post QC metrics to determine the resulting quality, because I think in the end, the data that comes out is really the final determinant of success with an FFPE tumor tissue.
Well, we should end on that. I think we've covered a lot of ground regarding tissue stewardship today. And I'd like to thank all of our listeners for joining us. And of course, to you both for your time. I'm so grateful that I get to work alongside you every single day. It's such a special opportunity to have some of the smartest people I know on our team. Until next time, we hope you will join us on Labcorp Perspectives Oncology.